Hello, this video is about embryology. Normally, the period of pregnancy is divided into three phases: germinal, embryonic, and fetal period. But for our sake of convenience, we'll discuss the development in weeks. This session will discuss about week 1 and week 2. So, three important changes happen in week 1, namely fertilization, cleavage, and implantation. Fertilization is fusion of two gametes. Transport of oocyte from the womb to the fallopian tube, and transport of spermatocyte from the cervix to the tube. And after once inside the tube, it will be penetration of sperm into the corona radiata, into the zona pellucida, and fusion of the plasma membranes. As a result of fertilization, what happens is the second meiotic division is completed, diploid number of chromosome is restored, and you can determine the chromosomal sex of the future individual to be born. A point on capacitation. It is the final stage of maturation of the spermatocyte that is happening inside the female reproductive tract. It takes nearly 7 hours, starts in the uterus and continues up to the tubes. It is believed to be because of calcium influx and certain release of chemicals from the sperm head, and the follicular fluid is said to promote this process of capacitation. The second event is cleavage. Cleavage is because of a repeated mitotic division of the zygote within the zona pellucida in rapid succession. So the zygote divides into numerous cells. All cells will be equal in size and each cell is called the blastomere. And this phase lasts for 6 days. This point within zona pellucida is important because zona pellucida is something that protects the underlying trophoblast. So if the zona pellucida is lost, the trophoblast will be exposed. The trophoblast is very sticky cell, and whichever is surface it comes in contact with, it will try to anchor and it will extract the nutrients. So imagine the zona pellucida is lost very early. What will happen? The trophoblast will stick into the fallopian tube and the ectopic pregnancy results. So intactness of the zona pellucida is important for implantation properly into the uterine cavity. And this phase lasts for 6 days. So compaction, it starts at 8 cell stage. They have outer tight junction and the inner gap junction. This is to facilitate the exchange of nutrients and transmission of electrical signals. Morula is 16 cell stage. Even at this stage, the zona pellucida is present. When the cell is in the morula stage, the outer cells will become differentiated into trophoblast and the inner cells differentiated into embryoblast. Blastocyst is a stage where the blastomeres will have a cavity inside them. It's called the blastocele, and at a particular point, the zona pellucida will disappear, and that is called as hatching. It happens on day 5 or day 6. It helps in implantation. So the third event, that is implantation, it happens 6 to 12 days after fertilization. The events happening in the uterus is the decidual reaction. Normally after ovulation, what will happen? The uterus will be in the secretory phase. But in pregnancy, because of exaggerated production of HCG and progesterone, the secretory changes are exaggerated. Hatching happens. The trophoblasts get adherent to the uterine epithelium. Compactum and spongiosum erodes, and the trophoblast will differentiate into cyto and syncytio trophoblast. Syncytio or multinucleated, and they have no mitotic figures. Cyto or cuboidal and mononucleated, otherwise called as Langhans layer. And the penetration defect will be closed by a fibrin coagulum. There are 3 types of implantation. Central is happening within the cavity, that happens in cows. Eccentric is where the cavity is having a nidus connecting with the main cavity, and the implantation happens in the nidus. Example, mouse. Interstitial is where the implantation happens inside the endometrium, and that is in humans. What is the decidua? The stratum compactum after implantation is called the decidua. Because of high HCG and progesterone, the stromal cells will enlarge and they contain glycogen and lipid vacuoles. The intercellular substance is increased and the vessels become more tortuous and they have high vascularity. So the decidua that is covering the part near the implantation site is called the basalis, outside is called the capsularis, and elsewhere it is called the decidua parietalis. So to sum up, in day 2 it is 2-cell stage, day 4 is morula, day 5 is blastocyst, and between day 6 to 7 implantation happens. Now we'll go to week 2 changes. The events in week 2 can be differentiated, can be divided into 3. That is the changes in the embryoblast, change in the trophoblast, and the endometrium. So in the endometrium, change is the same. The decidua basalis, capsularis, and parietalis. That's all. And the cavity fuses. Trophoblast is differentiated into in situ and cytotrophoblast. And the trophoblast helps in the formation of villi. Embryoblast is the main core, and they form the embryonic disc and the procaudal plate. In this image, you can see the green ones are the outer cells, that is the trophoblast, and the yellow and the blue ones are the inner cell mass. So the division into outer cell mass and inner cell mass is happening at day 16 itself. So the outer trophoblast is divided into syncytio, outer syncytio, and inner cytotrophoblast. So those in the light green are the cuboidal cells, mononucleated cells. They are the cytotrophoblast. The outer or the syncytiotrophoblasts, they are multinucleated and they have lacuna. The inner cell mass is differentiated into epiblast above and hypoblast below. This epiblast is columnar and the hypoblast is flat cells. Above the epiblast you have the amniotic cavity, and below the hypoblast you have the yolk sac. So amniotic cavity is bounded by cytotrophoblast above and the epiblast below. Yolk sac is bounded by cytotrophoblast below and the hypoblast above. This is the boundary. After the epiblast and hypoblast has been formed, what happens is among this epiblast, one particular point is having taller columnar cells. That particular point is fused with the hypoblast, and this forms the embryonic plate. It determines the polarity of the embryo. And this is the place where the procaudal plate also forms. So at approximately 12 days, what happens is this inner trophoblast, inside the inner trophoblast, there is a brown tissue called the extraembryonic mesoderm. It is called extraembryonic because it is outside the embryonic disc. So this extraembryonic mesoderm is divided into splanchnic and somatic mesoderm. So splanchnic is the one that is coming in contact with the yolk sac, and somatic is one that comes in contact with the trophoblast. In between these two mesoderm, you have a cavity called the extraembryonic cavity. So after this extraembryonic cavity expands, what happens is the primitive yolk sac will appear like reduced in size. Okay, this is called the secondary yolk sac. Yolk sac is named so not because it contains yolk or something like that. It is a site of erythropoiesis in fetus. Okay, and this is the villi of the fetus. So there will be a main trunk that is called the truncus corae. Branches, there are multiple branches, that's called the ramus corae, and these branches divided into small things, that is called the ramulae corae. Development of placenta we'll discuss in detail in the next session. But one thing you have to know is villi has primary, secondary and tertiary stages of development. So first thing that is happening is below the trophoblast you have a mesoderm, right? This trophoblast is differentiated to cyto and syncytio. The syncytio is having lacuna, and this lacuna gets enlarged and they form trabeculae. So in primary villi, there will be lacuna covering the core of cytotrophoblast surrounding syncytotrophoblast. In secondary villi, the mesoderm invades. In tertiary villi, there will be capillaries inside the mesoderm. So this chorionic villi, that is towards the decidua basalis, will form the chorion frondosum, and towards the decidua capsularis will form the chorion leve. This chorion leve is short-term and it will disappear because of pressure as expansion happens, and this frondosum will form the fetal part of placenta. So to sum up, in day 8 you have bilaminar disc, and by day 12 the implantation will be complete, and by day 14 you will have secondary yolk sac and procaudal plate. Thank you.